In this video, we're going to be distinguishing gram-positive and gram-negative bacteria and talking about the main things you need to know for the MCAT. So first of all, when it comes to bacteria, remember that most of them, not all, but most have a cell wall. And that cell wall is actually referred to as the peptidoglycan layer. It's called a peptidoglycan because it's actually a polymer of sugar and amino acids or peptides, okay? Now, as it turns out, when we do what's called a gram stain procedure, we can actually distinguish gram-positive and gram-negative bacteria by color, where the gram-positive bacteria, as you can see, will appear purple, and gram-negative bacteria will appear pink. And the reason for this is actually simply due to the relative thickness of the peptidoglycan or the cell wall, where if you look at this simple diagram of these bacterial cells, a gram-positive bacterial cell will have this relatively thick peptidoglycan or cell wall surrounding its plasma membrane, whereas a gram-negative bacterial cell will have its plasma membrane and then it will have a thin peptidoglycan right outside of it, much thinner than the gram-positive one, and then it will also have the unique feature of having another plasma membrane, an outer membrane around that peptidoglycan layer. Now, how does a gram stain procedure work such that we get these two different colors? Well, initially you have, let's say, a Petri dish with some unknown bacteria, right? Maybe multiple types of bacteria. And the first thing that we're going to do, as you can see here, is we're going to add in this crystal violet purple dye. It's basically a dye, okay? And what it does is it begins to try to enter the bacterial cells and it will start getting stuck in the cell wall for both the gram-positive and the gram-negative cells. The next thing that we're going to do is we're going to add iodine. And the reason we add iodine is actually simply to try to make that crystal violet purple dye stick to the peptidoglycan. And what I want you to remember is that the iodine is going to actually make it so that crystal violet gets stuck in the gram-positive cells, but not the gram-negative cells. And that's mostly because the peptidoglycan is much thicker for the gram-positive cells. The next thing we're going to do is the washing step. The washing step, we're just using alcohol. We add an alcohol to sort of try to get the crystal violet out. However, if it's a gram-positive cell with that thick peptidoglycan, the crystal violet is stuck very tightly, so it will not be washed out. But with the gram-negative cell, because of the thin peptidoglycan, the alcohol will successfully wash out the crystal violet dye. And so at this stage, the gram-positive bacterial cell will be purple if you looked at this point, but the gram-negative cells you would not be able to see. They would be transparent because they don't have any dye. And then the last step is we will add this sort of pink dye called safranin, and safranin will only get stuck to the gram-negative cell, the transparent cell. It won't get stuck in the gram-positive cell, and that will turn the gram-negative cells sort of pink or a little bit red. And then at that point, when we look under the microscope, we'll see two sort of main populations, the gram-positive, the purple, and then the gram-negative, the pink. Now, why would we want to do this? Well, imagine we have a patient that's very sick. They're in the ICU or just in the hospital, and we're suspecting a bacterial infection. We'll get blood cultures or urine cultures, probably both. And if we start seeing bacteria growth, right, we're going to gram stain, or we're going to gram stain right away, basically, and we're going to try to distinguish, first of all, these two major categories, gram-positive and gram-negative, okay? And this is really important because gram-positive and gram-negative bacteria have unique structural features. I mean, you can see that here with thick versus thin peptidoglycan and other things that will make us tailor our antibiotic choices differently depending on if it's gram-positive or gram-negative. Okay, so now let's zoom in on these different types of bacteria and actually look a little bit deeper about the differences between their sort of outer structure. Okay, so if we look at this, like this diagram, we're looking at the plasma membrane and then the peptidoglycan, aka the cell wall. And you can see that the gram-positive one, which is going to turn purple, right, well, it has this very thick peptidoglycan. And remember that this is just a polymer of sugar and amino acids, okay? And then, of course, it has its plasma membrane, which is really like the plasma membrane of any other organism like eukaryotic cells, where it has proteins, it has lipids in it, like phospholipids, okay? But if we look at the gram-negative bacteria, this one's a lot more unique because it has the plasma membrane, as always, and then it has this space in between this plasma membrane and this plasma membrane, which is called the outer membrane. And this space where we actually have that thin peptidoglycan is called the periplasmic space, okay? And this periplasmic space contains the thin peptidoglycan. And it's because it's thin that that initial purple stain does not get stuck and it ends up getting highlighted in pink instead, okay? Now, notice that it also has this outer membrane. Now, the outer membrane has a really important component that I want you to remember, and that component is actually called the lipopolysaccharide, which I'll abbreviate as LPS. Now, the lipopolysaccharide is also something referred to as an endotoxin. And it is extremely immunogenic. And what I mean by that is it stimulates very violently the immune system. And there is something in medicine that is called sepsis. Sepsis is a condition, basically it's a very dysregulated, violent response that the immune system exhibits to some sort of infection. And LPS is a major trigger of sepsis. In fact, we would call that gram-negative sepsis, which unfortunately is very, very dangerous, okay? Because the immune system can react very violently when LPS is present, okay? So that's called an endotoxin, aka lipopolysaccharide. Now, as it turns out, the gram-positive bacteria also has something, basically these antigens in its peptidoglycan, in this case, instead of an outer membrane, which are called teichoic acids or lipoteichoic acids. Okay, so I'll write that down. Lipoteichoic acids, or just teichoic acids. And really, I just want you to be aware that that's present. You might see that term. That is also basically an antigen, okay, for gram-positive bacteria, although typically it is not as immunogenic as the gram-negative LPS, all right? Now, the other thing is the gram-negative bacteria, you might see this will also have something called an O antigen, which again, your immune system can produce antibodies against and other adaptive immune cells can recognize, all right? So what are some major examples that you might hear about of gram-positive and gram-negative bacteria? Well, two major groups of gram-positive bacteria are going to be the Staphylococcus, okay, like Staphylococcus aureus, and then there's also Streptococcus, okay, Streptococcus, like Streptococcus pyogenes, which is a major cause of like sore throat, for instance, all right? Now, gram-negative bacteria, there's many more, of course, gram-positive, but those are two major groups. And then gram-negative, you might have, of course, heard of E. coli or Escherichia coli, all right, which is a major cause of UTIs and other things, all right? And then also there's something called Salmonella, which is a foodborne illness. And there's different types of Salmonella, of course, but those are some major examples. And you'll hear of many more, especially if you take a microbiology class and, of course, in medical school.